Welcome to Channel 17, the Town of Colonie Government Channel.
Barbara is the chair of the biology department at Union.
Former
right?
chair.
Former chair?
Yeah,
okay.
Just stepped down.
She's on sabbatical right now, working over at RPI. And for this book about cells, she teaches classes in the mechanisms of cell regulation, physiology of cells and organisms, and the molecular biology of the cell. So who better to talk about this book? The Immortal Life of Henrietta Lacks. So here's Barbara Danowski.
Thank you all for coming out for this on such a beautiful fall afternoon. Yeah, I'm a cell biologist and I love cells. I love looking at cells. And in fact, one of my courses has been renamed and the focus is cancer cell biology. And students learn about what's the difference between normal cells and cancer cells. And so this book, I love this book. I've worked with HeLa cells, and I know a bit about the history of cell culture and how difficult it was to be able to culture cells, to get cells to grow in culture for experimental purposes, and the importance of those cell lines, and there are many, many cell lines now, for current research. And so I was delighted when Joe Nash asked me to take part in this discussion. So I assumed that it would be more of a discussion, but some of you have not read the book, so I do want to give a very brief synopsis of the book. The other thing I want to tell you is if you have some time and you want to stick around later, I brought with me a little microscope and some toothpicks and sliding hover slips, and the cells lining your cheek are very easy to just scrape off with a toothpick, and they look very similar to cervical cells, okay? And in fact, many of you women have had pap smears, and that's exactly what the doctor does. He takes a little swab and he scrapes some cells off and looks to see if they look normal. And cervical cells will look a lot like your cheek cells in appearance. Only they make different proteins and things. So if you want to do that, I'll stick around and we could do that if you're interested. It's kind of fun. So the story, The Immortal Life of Henrietta Lacks by Rebecca Skloot. There are three stories here that I think of that I found just so compelling. The first one has to do with Henrietta Lacks herself, the way her disease, that she came down with cervical cancer. She was treated at Johns Hopkins. For those of you who don't know the book, I should summarize this. There's the tissue samples were taken from her cervix. 2 little dime-sized chunks of tissue were taken, one from the tumor and one from surrounding normal tissue, and brought into a lab because researchers there, that was the norm to do that. And there was a particular researcher by the name of Dr. George Gay, who was trying to grow cancer cells in culture. And those were taken from her in one of her earliest visits to Johns Hopkins. And subsequently, in the course of a year, her tumor grew uncontrollably despite the current treatments that were normal for that day, radiation treatments. They would actually sew a vial of radioactive material into her body. That was the norm to see if it would kill off the cells. And she eventually died. But interestingly, that little chunk of cancerous tissue, not the normal tissue, was able to grow in a liquid medium. And this researcher, that was the first time after trying this for many, 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 many samples taken from every patient that came through the clinic with cervical cancer, chunks of tissue were taken, and that one grew. By growing, I mean that the cells divided in culture So one cell makes 2, makes 4, makes 8, 16, and they perpetuated in culture, in a liquid culture. And that was a huge breakthrough. That was the very first human cancer cell line that was ever created, essentially. All right. So Henrietta Lacks, so that story, how that happened, and subsequently how those cells were used, and how her family found out that they were even in existence is the bulk of the story here. It's the main story. But I find there are 2 other side stories that I would love to hear your opinions of. The other one has to do with her daughter, Deborah. So Henrietta Lacks had 5 children. One of them, her oldest daughter, was born with some, they diagnosed it as idiocy then, and she was put in a home, in a state-run home, okay? And, but Deborah was her second daughter, and Deborah was only 2 or 3 years old, I believe, when her mom died. And she never knew anything about her mother. And the quest that Deborah undertook to find out something about her mom is intimately tied into the book, and she put all of that energy into trying to understand something about her mom's cells. Okay, so that her mother's cells were immortalized; they were growing perpetually in culture, gave her a connection to her mother that she never had. And I think that that's just such a strong human emotion to want to know about your parents. So adopted individuals wanting to know their birth parents, wanting to know where they came from is so very, very important. And so a lot of this story is about Deborah's quest, okay, and how the author helped her, first had to build her trust, of course, and then helped her to understand her mother through her cells, understand the cells. How did she find out about the cells? I should say that I gave you the timeline. There's a lot of information on the web if you look up Rebecca Skloot, the author. Henrietta Lacks's cells were taken, and the first cells were cultured in 1951. But the family found out essentially by accident about the cells somewhere in the mid-1970s. This timeline, and I'm more inclined to believe this one, is 1973. The Lacks family learns for the first time that Henrietta's cells are still alive. There was a television documentary made in Britain by the BBC that was called The Way of All Flesh, and they give the date as 1976. So I'm saying mid to early 1970s is when the family found out, in fact by accident, when researchers were sent to find the family to get blood samples because they wanted to know more about this particular cell line. So Deborah's story, I think, is an interesting one to consider here if we want to have a bit of a discussion about it. The third story that I find really interesting is a story of the author, because there have been stories written about the Lacks family and Henrietta Lacks. There was a story in Rolling Stone and some other newspapers, and there was this BBC special that interviewed the existing family members, many of them. But this particular treatment that Rebecca Skloot gave to uncovering really what happened and really getting to know this family on a personal level, I think is something special. And just last night, in fact, I was thinking about this book for a long time and what I would be saying about this book, but it was only last night that I got on the web and looked up all this information. I didn't realize there's enormous wealth of information, discussion questions, and all of that. But one thing really leapt out at me, And it's a story about the author. And I don't know if many of you have read this, so I'm going to read a little bit from this, and this is directly from the website. And it's on the second page. You don't have this, but you can look it up. An interviewer asked Rebecca Skloot, why was the existence of the HeLa cells so difficult? Oops, that's not it for Henrietta's family. Sorry. I can't find what I was going to read. So the story is this. When in the book, and many of you have read it, the author says that she was in a high school biology class, and her high school teacher threw out a very simple statement and said, yes, and there is a cell line called HeLa cells, cervical cancer cells, and they were and they originated from a black woman, and I think that was it that they said. Do you remember something like that?
I don't think he even said that where they originated. They just talked about the cells.
He did say that, and she was black. It came from a woman, Henrietta Lacks, and she was black, and 1950s perhaps, something like that. And this struck Rebecca Skloot. It turns out that from high school and then I guess a little bit later, her father developed some sort of odd disease that they attributed to a virus. And he lost the ability to walk, his thinking was impaired, etc. And he was enrolled in essentially in a clinical trial to try out some treatment for this. And she didn't say what the disease was, and she didn't talk about the outcome, but she said that she brought him to the hospital for his treatments and sat there with him because he couldn't drive and drove him home, etc. And at some point, the trial was disbanded, essentially. Whether it was from lack of funds, etc., it wasn't really said. And so she had sort of a first-hand experience with what it's like to be a subject in a research study through her father. And she saw what kind of information the subjects are given and what information they're not given. She did go on to major in biology as an undergraduate before she went on to graduate school and focused on writing. But that, I think, played a strong role in how she approached this story. Her empathy for the family and for wanting to help the family understand the process that had happened, what had happened to their mother, and and all of the miscommunication that occurred throughout the time from the moment that the family knew of the existence of the cells. And again, remember, it was 20 years later that they found out that this cell line existed. Okay? And so those three threads, I think, were so wonderfully woven together to make this such an enjoyable read. But there's also a lot of disturbing things about this book, right? Now, for those of you who haven't read it, what background information can I give you about this? A few more facts, okay? So this cell line was created. Researchers used it. The researcher whose lab was focused on trying to get human cells to grow in culture, from what everybody has said, was doing it purely to hoping for someday to contribute to a cure for cancer, okay? He did not sell the cells for a profit. He, in fact, took tissue samples from himself, from his children, etc., to try to get them to grow. He spent some of his own money in lab supplies to keep his research running. So, nevertheless, Dr. George Gay did not tell Henrietta Lacks that her cells were growing in culture. And Henrietta Lacks was still alive when he knew those cells were growing in culture. And as a scientist, I find that troubling, right? I mean, George Gay was not trying to make money off of this, and he was sincerely interested in contributing to the greater good. And he received a lot of fame and notoriety among fellow scientists for the development of this cell line. But the development of the cell line has to do fundamentally with the defect in those cells that had been normal that were somehow changed and altered to become cancer cells. It's really not anything necessarily that he did, or for that matter, that Henrietta Lacks did, right? But he was able, through his efforts of taking sample after sample after sample from patient after patient after patient, was able to get these cells to grow in culture. But what I've never understood, so what bothers me about this, is why didn't he tell her? And the samples were taken before she died, and I believe the cells started growing right away. Why couldn't he tell her before she died, we have your cells and they will contribute wonderful things to medicine? And that just bothers me.
I want to make a couple of comments. First of all, I've actually worked with HeLa cells, believe it or not, at Johns Hopkins. And I did my postdoc there, and I published quite a few papers on work on HeLa cells. And believe it or not, this was in the mid to late 80s, early 90s. I didn't actually know her name. I knew that HeLa was someone whose name started first name H-E, last name L-A, but the lore was always it was Helen Lane.
Right.
And they mentioned that in the book. So even at Hopkins, at least among many people, her name was unknown.
Right.
And they are probably the most used cell lines still in the world. There are thousands and thousands of papers published using HeLa cells. We were using them simply because they grew very well.
Right.
We weren't specifically studying cervical cancer. As far as your point, I understand. One thing people need to know is that standards change over time. So at the time in 1951, there was the standards of patient confidentiality, patient rights to know was very different from what it is today. There really wasn't much, and a lot came because of experiments that were done by the Nazis, as well as the Tuskegee syphilis studies and so on. And so a lot during the late 50s, early 60s, etc., a lot of those standards changed. So I don't know that, so they certainly didn't act illegally in terms of the standards at the time. Even today, if you have a sample taken as part of a study, you do have to sign a consent. Mm-hmm. But I don't even know today that it's standard that you would be told, oh, your cells are growing.
No,
yeah.
it's not
even
standard
today, I don't, and
no.
I've been involved in studies where we've taken samples from patients, and it's just not one of those things that maybe you say it should be, and I wouldn't disagree with that. But it's just not sort of normal procedure. Mm-hmm. Now, today there would be specific consent forms and so on. But I don't know that the patient would have a right to call you and ask. Right. And then if they did, you would certainly answer them. Mm-hmm. But that you would sit down, find the patient's phone number, call them, I don't know. Gay was not her actual doctor.
Right, Jones was her doctor. Jones was her gynecologist.
So,
yeah.
I understand where you're coming from, and it's a close call.
We're all guinea pigs, basically. Whenever we go to the doctor, we're guinea pigs. And as far as research and that, because we don't give consent for them to just do research on any part of our body.
Oh, you have to give consent. And if you don't give consent, they can't take. They can take the sample, do whatever clinical test, and then they must destroy it. And those are respected, absolutely.
Right.
If you give consent, would you be told, 5 years later, oh, we published a bunch of papers on your tissue? I mean, there's no law says you would have to, and it
So
might
from-
be nice if you did.
From my perspective, though, and some of this is influenced by the fact that I watched the BBC documentary made called The Way of All Flesh. And they interviewed, well, I take that back. They had a little clip of George Gay standing up describing the cells, and he had a flask and how excited he was that he had this cell line at a scientific meeting. And I should say that I think your point is well taken. Scientists work on particular things, and often there's not a lot of people that you can share your excitement with that will understand it, right? So you share this information at scientific meetings. You know, for example, when I got my PhD, my mother told all of the relatives that I was a microbiologist because she knew I used microscopes, right? And I thought that was very sweet, but I couldn't really go into great depth of exactly what I was working on because there's a handful of people in the world that care. And so George Gay, I wanted so much, I thought that Rebecca Skloot did a lovely job of not laying any blame really on too many people, just sort of laying the story out. And I really wanted to like George Gay. And my saying that, why couldn't he have told her, comes out of the fact that he was so excited about this. Those cells grew. And there were no norms, as you said, back then. There was no obligation. But yet, he had devoted his life to this. You would think you might want to say it. Can't fault him for it, but boy, I wish he did, right? She died within the same year that the sample was taken, but the cells grew immediately.
Yeah, keep in mind that other cells grew for a time.
Right.
And then they ran up against
right.
their...
There is something, I should say, too, that before I went to graduate school, I worked at Sloan Kettering, and one of my jobs was to take tissue samples of tumors, mostly melanomas, and to try to get them to grow in culture. And patients gave consent because the idea was these cells were used. If their cells grew, they were grown in culture, and they were used, they were modified, and patients in this study where their immune system was they were trying to stimulate their immune system to recognize their own cells, and they were injected as a vaccine back into the patients, their own cells. And that was part of this study back in the late 70s, early 80s. But there are some tissue samples, some tumors, and when they grow like flop. And there's just no denying it. And I believe that was the case with the HeLa cells, quite frankly. I mean, they noticed his lab tech, the person who did them, I believe Mary Kubitschek, said right away she was so surprised that they grew pretty quickly. But yes, I don't think we can, there's no one to blame. Everything that was done was the norm for the time, okay? Nobody in their wildest dreams in the 1950s and 60s ever thought that people could sell these cells or people could own genes or DNA sequences. And we're in a very different world now, right? So back then, that it was the norm, okay?
I disagree. When a scientist takes a specimen from anyone, he takes it from the person.
Right.
It is not his. He should explain everything that he intends to do with it, and in the future, go back to the person and ask, can I do this with your specimen? And if people, the researcher, don't think of people sometimes as human beings. They think of the goal they're going to get to. Look what happened in Nazi Germany. They didn't intend to do what they did, but they did it.
Right.
Because they had no,
Tuskegee studies and.
they had no
Yes.
control. A person who's going to win the Nobel Prize or whatever don't practice control unless they're that way naturally. So they should not have done it. That's my opinion.
Yeah.
Sorry, but are we talking about this case specifically, or what should be done today?
Right.
You know, that I guess is a big question.
Right. I think we have to. I think what happened. I mean, yes, it's
Can I speak
egregious.
for myself?
Yes.
Human nature doesn't change. The way Adam and Eve was, the way we are today. Human nature doesn't change. Technology changes. You may think I can do something with DNA confirmation or whatever, but the person who's in charge of it can be evil or good with the techniques.
Maybe he did it to protect his research. If he shared it too much with too many people, it might have foiled what he was trying to accomplish. I understand that times were very different and things have changed tremendously. That's why we have patents now to protect what we've created. So maybe he did it in defense of the doctor. Maybe he did it to protect what he was truly discovering in turn to help all people.
To get back to this gentleman's point, we would like to think we own our tissues and that people should not take things from us and use them without asking us. But there's a long, long history of, this sounds horribly gruesome, I'm sorry, cadavers being dug up and used for medical studies back in medieval times and in 1600s, 1700s, 1800s. That's how people learned about the body and medicine, etc. And so I'm not saying that it's just there's been this slow evolution of patients' rights. And I think that that's a good thing. I have trouble as a scientist condemning the Hopkins people because they were doing what was the norm at the time. What I find really offensive, and I'll move on a little bit, is that when the Laxes found out about the cells, which was in the 1970s, Deborah in particular, and tried to find out more about what that meant. What are cells? Can you tell what my mother smelled like? Can you tell me? Right, she clearly didn't know or understand cells, and why should she? And one of the doctors who was interested in getting blood from the family members so they could do genetic studies and other studies on the family and map certain chromosomes, handed her a textbook of genetics and says, this is going to answer your questions. And that absolutely just put me over the edge. That was so insensitive. Likewise, even in the 1990s, the BBC people, Adam Curtis was a scientist, and he spearheaded this BBC special and interviewed Deborah. And you can watch this on the web, by the way, The Way of All Flesh. And if she wandered off from the storyline that he wanted, he would steer her back. She wanted to know about if they could clone her mother's cells, could they put it into an egg, and can she give birth to her mother? Things like this, and he kept steering her back to, what do you think cancer is, and etc. And so there was an insensitivity, I think, to the family once they found out about this that I think it's hard to excuse. I really feel that's hard to excuse.
I agree with that, and there's no call to be insensitive in that situation. But just to disagree with the gentleman over there, as a scientist, it's for example, what happens when a scientist develops a cell line that other scientists see as very useful is they immediately request that cell line. And that cell line gets sent all over the world, literally. And a cell line like HeLa is probably in thousands of labs all over the world. Mm-hmm. So the idea that every one of those people, before doing an experiment, needs to call up the patient who is dead, or call up the patient's heirs and family and say, may I do this particular experiment, is just not feasible in the real world.
I'm going to play devil's advocate here a little bit, just because as a scientist, I agree with you. Science is built on prior discoveries, right? No one person makes a breakthrough. I mean, I shouldn't say never, but mostly you are building on the knowledge acquired by other people, okay? And in general, I would like to believe that scientists are generous with their discoveries and their contributions. There have been examples when that's not the case, when people are selfish or their egos get in the way of their behavior, right? Where they will not share certain things, etc. But to get to your point that wouldn't it be awful if every time you wanted to use HeLa cells you had to contact the person? What about rights to songs? Anybody have children in high school productions of musicals? You want to sing those songs? Don't you need permission from either the estate of the person who wrote the song? I mean, it seems...
Yeah, but there is a mechanism whereby that's granted.
There is a mechanism whereby it's granted, and we don't have that mechanism in place now. And so maybe that's what we should... I don't know. I would find it a nuisance, but you know what?
If you're saying should the heirs of Henrietta Lacks be given, a small fee every time HeLa cells are used, that's another question. I mean, the courts have said they're not entitled to it. That may or may not be a right decision, I don't know. But certainly to have a scientist call up. I mean, I've done work where we've taken samples from hundreds of patients. And the idea that I would have to call each one, find where each one of them is, many are deceased, I would have to find a relative, I mean,
it would be hard.
First, this would be the equivalent of somebody working for, let's say locally, GE, inventing something, even on their own time, which they'll have to prove, or for the company. They immediately sign away their rights on going to work for any organization if they've invented something. So nobody seems to have a problem with that, okay? And even if the man or woman says to their spouse, well, I'm working on this thing, but it has nothing to do with GE, it has nothing to do with Union Carbide, it has nothing to do with any of these things. Okay, so it's the same thing. It's coming from an invention. An idea is coming from somebody's mind, which is no different than the cell. And that it's somebody's cell. So that's number one. Number two, which kind of relates to it, what I'm hearing throughout this is something that everybody's dancing around, and that is somebody's going to call their lawyer, and what is it? Bottom line, what is it worth? How many shekels is this going to pay me? And nobody's mentioning that. So I figured, all right, I'm going to bring it up. Because, one says, well, maybe we should have told him. And why? Why would you tell a dying woman who's in pain about this? And maybe he feels she wouldn't understand it. Not because of her education or her color or anything else, because many times researchers are insensitive people. Like a good deal of the population. So, as doctors are, I mean, who do you want for a doctor? Somebody with a great bedside manner or somebody who's really good? So the problem is not so much that they didn't tell her, but why did they tell her? Because people act they think they're acting rationally and logically, when in reality, they're operating on their egos, they're operating randomly. What they think are objective results of science, and I'll just leave it at that.
I cannot agree with what she said. I think the financial quotient comes in and muddles everything up. But also, I think what we really need to focus on is the big picture. And I know we're talking about the ethical issues surrounding it. And let's just leave Henrietta Lacks out of it just for what I'm saying. But in this day and age, you get informed consent, which means that you're saying, yes, you can use my tissues, my blood for research. And you can say no. But if you have said yes, then you are contributing in some way towards a cure for so many different diseases. And researchers are insensitive people. I'm a cell biologist myself, and I have worked. But we hope our primary goal in some way is to contribute, and with great thanks to the people who give us the samples, to something to find a cure for 80 different types of cancer. So I think we need to think about all the different implications before just considering one single ethical problem.
What solutions did they use?
What solutions did they use to grow the cells in? Okay, and then we'll get to your comment. So that's the other contribution that the HeLa cell line made is that people were trying all different sorts of things to grow cells. And I believe what George Gay did was he took clotted chicken blood or serum. I think he formed clots and grew the cells on the clots. So it had to be fresh. And there's a picture, I think it was in maybe one of those other, the Wave All Flesh, of him holding a chicken up and draining the blood from the chicken. Pretty gruesome. People tried all sorts of things. Currently, there's got to be some sort of what we call growth factors to help the cells grow, and the most common used source is serum from the blood of certain animals, preferentially fetuses, so cows. So the meat industry, if they slaughter cows for meat, they will collect the fetuses, and the blood gets sold to scientific companies that then sell it to us to grow cells at great expense, and other things. But they were trying many, many, many different combinations of salts and solutions and nutrients to see what is the best medium for cells to grow in. And HeLa cells provided that as well as a way of testing for what is the best combination of things to get cells to grow. Going back to the norms of the time, and for the record, the Nuremberg Code of Ethics, which is on this timeline, was 1947 that was established, and this is 1951. But the Nuremberg Code was not a law, but it made people cognizant of the fact that people should be asked if their bodies, tissues, etc., are going to be used. It was not adopted as a law, but I do think that the story of Henrietta Lacks really cemented the need for laws, and that's why we have laws in place now. I do think that at the time, doctors were on a pedestal, right? And there's a little bit of, for lack of a better word, intellectual elitism going on here. The extent to which there's some level of racism going on here is we have to acknowledge, I think. Gender issues as well. I mean, all of those come into play. And getting back to that notion of this intellectual elitism, I wanted to bring up Deborah for a minute. And I found myself really conflicted about her initially, but felt so strongly for her desire to want to know her mom, right? How really somehow needing, for her own self-esteem and for her own grounding, to feel connected to her mother. But I realized that she was outrageously brave. She asked doctors questions. She was not afraid of coming across as foolish. Her desire to know, what are these cells? What can they do? How are they being used? She was in no way inhibited from asking doctors questions, and some examples in the book are so naive you would think, oh my gosh, how could anybody think that? But she asked them because she was brave, and it mattered to her so much to know about this, and that he took the time, and that Rebecca made it her mission to make sure that Deborah and anyone else understood what was going on. And I think that was really important, and I think that came from Rebecca's own experience with her father. And I've come to realize how very brave Deborah was, really. Not being intimidated by this group of highly intellectual researchers. I thought that was amazing. That's right. Mostly white people, exactly. And she did it. She went forward. And I loved, towards the end, when she was hunting for information about her sister Elsie, how she referred to Rebecca as her reporter. Get your own reporter. And that Rebecca empowered her in a way that she hadn't. She had felt so helpless before. And I thought that was a wonderful thing.
Let me follow Deborah's lead and ask some really dumb-headed questions. Were these?
No dumb questions.
Was this the first time any cells had been grown in the laboratory, or the first time any cancer cells had been grown? What's the significance of that happening?
Okay, so first of all, there's no such thing as a dumb question. So it doesn't exist in my book. Every question's a good one. That's a good one. There had been some animal cells that were able to be grown in culture, and people had started doing this in the early 1900s, and I think there's some indication of getting cells to grow, meaning they divide, they undergo cell division, and one cell becomes two. Even if they eventually die out, which most normal cells will, they're limited in the number of population doublings, I'll use that word, where one cell goes to 2, goes to 4, goes to 8. And so animal cells there was some success with animal cells, and I believe there might have been an animal cell line. And when I use the word line, that means that they've sort of overcome that restriction that the cells simply die after a set number of population doublings. So the HeLa cells were the first human cell line, immortalized human cell line. So they never showed that limitation. They didn't die in the petri dish after 30 population doublings or 40 or 50 or something like that. They kept growing, and they still grow.
Do these cells ever differentiate into other kinds of cells?
So a lot of experimentation has been done on HeLa, and in and of themselves, no. And you've worked more with HeLa probably than I have. Are there, as people have learned more about cells and how cells work, there are some cell lines that if you treat them in a certain way, they will differentiate or develop into tubules or structures or produce different things. I don't know if that's the case with HeLa cells because I haven't worked with them in that capacity. I've grown them because they grow fast and
Yeah, I mean, that's one of the things to point out in terms of what's special about them, even to this day, is how fast they grow. And the book had a couple of chapters where they talked about a problem that arose, I believe, in the 1980s, where people were doing studies.
Up until the early 80s.
Yeah, people were doing studies on other types of cells, and they were seeing all of these results that were very interesting. And it turned out that all of these, or almost all of these cultures, were actually contaminated with HeLas. And what they were unknowingly, and the reason for that is that, you know, if you put in 10 million of some other cell and one HeLa cell, it grows so quickly that it will, in a fairly short period of time, take over the entire culture.
Right, there was
So there were
accidental
all of these
contamination.
papers that had to be called into question because it was likely that what they were looking at were actually healing. And that's why they asked for blood from the family members, was they were looking to develop ways to look at any cell culture and say, because it's hard to tell just looking at a microscope. So they were looking for actual chemical, biological tests where they could say, this is healing or not healing.
Do they know why Gila grows so quickly?
So there are a couple of, so the short answer is no. Cervical cancer cells, so Henrietta Lacks' cervical cancer cells were infected with a virus called human papillomavirus. And human papillomavirus is a sexually transmitted disease. And as if it's not in the book, it's in some of the supporting materials. Probably every adult is probably going to have some infection with a strain of human papilloma virus. There are over 100 human papillomavirus strains, and a very small subset of them are now believed to be the main causative agent for cervical cancer. Okay, so there are certain strains that will, when they come in contact with cervical cancer cells, the virus alters the DNA. I'll leave it at that. And those cells now are able, they lose their ability to control their cell division. And so normally cells will grow and they form a sheet and then they stop. They don't undergo a cell division. And they lose that control if they have certain genetic material from small number of strains of this virus. I believe they found that the HeLa cells had multiple copies of a particular strain of human papillomavirus. But even that, if you take normal cells and you put in multiple copies of this HPV strain, you don't get cells that grow to the extent of her cells. So that's why I say the answer is no. That it's tied up with HPV infection and numerous HPV infections, possibly different strains, and it's the combination of strains. And alterations in her DNA that made them especially robust, for lack of a better word, in terms of their ability to divide and produce more cells in culture. And one of the questions that I had to throw out for discussion is, would this book have outraged me and other people as much if no one had profited financially from it? And I think yes, that affected me. That some people have profited monetarily from this is irksome, especially since the family, as is pointed out so frequently, are struggling financially, can't afford health care. I think it speaks strongly to our need for, I'll get my politics involved in this, in universal health care. I mean, nobody should have to do this. How microbiological associates got involved in this, I don't know the story there. It could be something like, so for example, the Tuskegee Institute grew up huge amounts of HeLa cells so that polio vaccines could be tried out and worked out, and so they needed massive numbers of cells for this. That was a not-for-profit organization. My guess is their funding came from, at the time, I don't know, I'm assuming government funds. Maybe some, I don't think private foundations did much by way of donating back in those days, although I could be wrong. And then it got taken over by Microbiological Associates, a for-profit company. Is it because the government pulled funding? I mean, we know that happens a lot. And so rather than let the cell line die out, maybe that's what happened. I have no idea. But clearly, money was made. And this is going on a little long. I found myself thinking, what else of ours is used for profit? Now, please don't I was trying to think of examples, and I thought of this. And I live in Schenectady County in the town of Glenville, and every year I get a little brochure that I could buy trees, seedlings, and things, right? I can buy compost, okay? And it says don't use it on vegetable gardens, but you can use it on flower gardens. You know why? Because it comes from the sewage treatment plants, from our waste. So I'm thinking, I'm paying money for something that's mine, but it's not mine. I mean, and I'm thinking, but it isn't mine, it's been altered, right? It's now a good product. Somebody invented this process by which they could take our raw sewage, let's leave it at that, and compost it, mix it with bacteria, etc., and create a product that we can use. And isn't this environmentally sound and wonderful and everything? But what if a person who worked in this sewage treatment plant isolated a strain of bacteria just out of the blue that could eat sludge from oil amazingly, and they sold it to a company, etc. Does he owe me money? So, in other words, the intersection of what's yours and what's science and who owns what and who should profit from things, these are real issues that we're facing in a way that we never anticipated. I don't think we're ready for this yet, but we're confronting it, right? And I think that these are serious issues that we need to address. What's yours and what is not? What happens when you take something and you put the work and effort into it? If you see a way of making money from it, is that okay? Or if it comes from a human being, maybe it should be for the greater good. It should all be not for profit. Is that feasible in this day and age? I don't know. I think in the scientific community, certainly in the cell biological community, there's been a lot of discussion about forming collaborations with industry, with research, for-profit companies, and basic science research, because it can allow, because they have so much more money, to move things forward more quickly. But there's a price to pay for that. And where do you draw the line? And I think that these are really interesting things that we need to consider, and that this book brings that to the forefront. I think the book also brings to the forefront the need for us to really reconsider healthcare, honestly. After I worked at Sloan Kettering, I moved to Germany, which was West Germany then, and helped set up research lab. I brought piles of human cancer cells with me on the plane and helped set up a research lab with a German doctor that I worked with there. And I had universal health care. A lot of money was taken out of my paycheck on a weekly basis to pay for it, but I didn't care. Anything I needed, it was just a given. And I miss that. And yeah, it's very expensive, but I think we're worth it, personally. I think if we have to decide where to spend money, I think that should be for that. Anyway, that's my personal point.
There's some issues that we're bouncing over. We haven't hit right on. You started there. I thought you would hit the word values.
Yeah.
But we haven't heard that word here.
Ethics.
You bounced around that, left and right, top and down. We haven't hit that, right? And the other comment I have is that I know many of us, I'm sure here, have arranged to make an organ donation on our passing. Mm-hmm. Now, I'm not going to be compensated for that. My family's not going to be compensated for that. I feel it's the right thing to do. Right. To let someone else enjoy that, all right? And conversely, maybe I'll be the recipient of a kidney or something someday. I don't know, all right? There's no money coming out of me for that directly, all right, for donation. But is that a similar issue to what's happened here?
Yeah. Is it a similar issue, organ donation and not getting money for it? When you hear about companies that go to countries that are in desperate dire need of money, where they pay people a pittance to remove a kidney, etc., these black market organs, it's clearly an issue, But I do think you're absolutely right, values or the ethics of things, and to develop what is the right thing to do. And it's hard, I think. I mean, I view, I do basic research, and I think of just increasing knowledge for the fun of it, for it, and I never think about profiting from it other than just trying to learn more. But there are people whose goal is to make money, right? And pharmaceutical companies, I don't want to go there because I don't know enough about how pharmaceutical companies work, but in order to pursue the development of a drug, for example, they do an analysis, a cost-benefit analysis, and they're not going to pursue something that isn't going to give them some level of profit. Or if they do, they have to be making a huge profit somewhere else to spur that on. And is that ethical? I mean, how do we separate? You know, we've got the business interests where the goal is to make a profit, and you've got
Similarly, what we see today, is the universities have set up separate corporations for technology commercialization. All right. So who's profiting from that? I mean, many people here may be very surprised to learn that many of our major universities hold a lot of patents and secure a tremendous amount of money every day of the year from selling those patent rights to do them.
Oh, yeah.
Absolutely.
I've worked in the biotech industry.
I mean, it's
Absolutely.
enormous.
Because if you didn't get a patent, then someone else would patent it and make money off of it. So the university says they would like to make money off of it so that, and I, being naive and idealistic, say it's so that they can fund researchers to do basic science who can't get government grants.
I'll tell you from my time in the biotech industry, when I negotiated with other companies versus negotiating with universities, the universities usually asked for more. I swear to God, we had easier times negotiating with other companies than with many universities. They wanted the sun, the moon, and the stars. And the companies tended to be at least realistic about what they wanted.
Why don't you sum up the book, and if you have more questions, Barbara will be here if people want to come and ask her first.
Right, and if you want to look at cheek cells, we can do that too. Yeah. What can I say about it? I think that this book came at a really wonderful time. I think that we need to face these issues of who owns the bits and pieces of us. Are we happy with the laws that are in place? How can we further science, and also who's going to fund it? I mean, I do think that we're sort of stuck with collaborations with profit-making organizations and not-for-profits, but how? I think we need to work that out, and I think that this book highlights that. I also think this book highlights, with respect to the Lax family, I think that it's about time that they learned, that they were given the chance to fully understand the contributions that their mothers and grandmothers, great-grandmothers' cells made to human health issues, right? And that it will be in textbooks now, I bet, right? And that's where it should be. And I think that Rebecca Skloot did an amazing job with the book. Other people have told the story of Henrietta Lacks, but not like this. And so I think it was a phenomenal read, and I think it's going to be around for a very long time. I think.